drive to stay focused, to push through resistance. It turns out that's not just mental, it's biochemical. New research is revealing that your brain's capacity for motivation may hinge on how well it handles oxidative stress. And at the center of that defense is glutathione, your brain's master antioxidant, at least one of them. In today's episode, we're exploring how this molecule fuels motivation at the cellular level and how nutrition, supplementation, and simple daily choices can help you sustain cognitive effort when it matters most. This is Daily Value, and I am William Wallace. Motivation is our internal process that compels us to initiate, maintain, and successfully complete actions directed toward meaningful goals. It's the physiological force enabling us to navigate challenges and overcome the inherent cost of effortful tasks. Motivation essentially governs how effectively we utilize our attention, memory, language, and physical actions to achieve rewarding outcomes. In essence, motivation allows us to coordinate executive functions to achieve meaningful goals. Our capacity to remain engaged in challenging tasks, whether they involve long-term work projects, academic pursuits, or physical exercise, is deeply dependent upon our motivational state. It shapes not just our immediate actions, but also our broader life achievements and well-being. The brain tightly regulates motivation through complex neural circuits. Central to this process is the nucleus accumbens, a region located deep within the brain's reward and motivation circuitry. The nucleus accumbens collaborates closely with other brain regions, including the ventral tegmental area, prefrontal cortex, and the amygdala. This interconnected network constantly assesses the value of potential rewards, calculates effort costs, and directs decisions about whether to pursue or abandon specific actions based on their perceived worth. Incentives energize effortful behavior through recruitment of the nucleus accumbens. The neurotransmitter dopamine is produced primarily in the ventral tegmental area and plays an essential role in energizing these networks. Dopamine neurons facilitate the assessment of motivational value, effectively energizing and guiding behavior toward rewarding outcomes. It is our motivational state that determines how we employ attention and working memory to tackle complex or demanding tasks. The motivated control model explains how under demanding conditions such as prolonged mental or physical effort, we mobilize cognitive resources strategically. When task goals are perceived as important enough, our brains channel additional cognitive and physical resources to sustain performance despite growing challenges. Importantly, motivation is not constant. It fluctuates continually based on internal physiological states, even when the actual difficulty of a task remains stable. Our subjective willingness to continue exerting effort can significantly decrease over time. This variability shows the dynamic nature of motivation as individuals frequently take breaks or cease activity altogether once their internal motivation diminishes. Fatigue strongly influences our capacity for sustained effort. Fatigue, commonly experienced as brain fog, frequently accompanies declines in motivation, especially after prolonged cognitive or physical effort. Fatigue manifests as a growing sense that the continued effort required to achieve a goal no longer feels justified by the potential reward. But how exactly do we become fatigued? Well, there are hidden determinants of fatigue that underlie persistence in the face of effort. At a physiological level, fatigue appears intimately linked to oxidative stress, an imbalance arising when excessive reactive oxygen species generated by intense brain activity overwhelm the brain's ability to neutralize them. You see mitochondria meet the high energetic demands of the brain during demanding cognitive effort. Elevated levels of oxygen in the brain is one of the prices we pay for elevated neuronal activity. Reactive oxygen species are produced naturally as a byproduct of ATP synthesis or the brain's energetic metabolism, particularly during effortful activity. If unchecked, these reactive molecules can impair neuronal function, leading to cognitive and motivational fatigue. So how do we protect neurons from oxidative stress? That's where one of the brain's chief and most potent antioxidants comes in, glutathione. Glutathione is a tripeptide composed of 3 amino acids, glutamate, cysteine, and glycine, of which cysteine is the rate-limiting substrate Glutathione is synthesized through a highly regulated enzymatic pathway, and its primary role within the central nervous system is to maintain redox balance and mitigate oxidative stress at the cellular level. Unlike several other antioxidants, glutathione is uniquely positioned intracellularly, acting directly within the neuronal and glial cell environment to neutralize reactive oxygen species at their source. While glutathione's broad antioxidant role is well known, its relevance specifically to motivational processes, particularly effortful behavior, is only starting to emerge. To reiterate what I said earlier, elevated neuronal activity, such as that observed in sustained effortful tasks, triggers increased oxygen consumption within mitochondria, specifically within motivation-related brain circuits like the nucleus accumbens. This heightened metabolic activity inevitably produces reactive oxygen species, which, if inadequately neutralized, can impair neuronal function, compromise cellular integrity, and diminish motivational capacity. Importantly, glutathione uniquely mitigates this risk by directly neutralizing reactive oxygen species within neurons, particularly during states of increased energetic demand, thereby protecting the integrity of cellular components critical for maintaining the neurotransmission responsible for motivation. Accumulating evidence now supports the hypothesis that glutathione levels in the nucleus accumbens directly influence the capacity to sustain effortful behavior. Clinical observations have noted altered glutathione concentrations within motivation-linked brain regions of individuals suffering from motivational deficits, including disorders that are characterized by apathy and anergia, which is an abnormal lack of energy. The available evidence points to glutathione as not merely a general protector against oxidative stress, but as a targeted mediator of motivation, acting specifically by preserving neuronal function under conditions of increased oxidative challenge. Understanding precisely how glutathione availability impacts motivational performance, however, requires deeper mechanistic insights. A recent study has made significant strides towards this understanding, revealing how exactly fluctuations in nucleus accumbens glutathione levels might shape an individual's motivational capacity. A study published in 2022 by Zalakouros and colleagues used high precision brain imaging techniques known as proton magnetic resonance spectroscopy. The researchers first measured glutathione concentrations in the nucleus accumbens of healthy human volunteers. Participants then performed an effort-based and motivational-based task, which required sustained effort, specifically gripping a sensor at half of their maximum strength to earn monetary rewards. The results revealed that individuals with higher nucleus accumbens glutathione levels sustained their performance consistently over time, particularly when high-value rewards were at stake. Conversely, those with lower glutathione levels showed a clear decline in their ability to maintain effort, especially during the later stages of the task. To establish whether glutathione levels directly influence motivation, the researchers then replicated and expanded upon these findings using controlled rodent models. Rodents were trained to perform progressively challenging tasks for rewards, and then researchers carefully manipulated glutathione levels directly in their nucleus accumbens. To achieve this, they used an inhibitor drug that selectively disrupts glutathione synthesis. When nucleus accumbens glutathione levels were experimentally reduced, rats rapidly lost motivation, quitting tasks sooner and showing lower thresholds for giving up. However, when glutathione levels were boosted through nutritional supplementation, specifically by providing N-acetylcysteine, also known as NAC, a well-known glutathione precursor, rats displayed significantly improved endurance, working harder and longer to obtain rewards. This intervention produced approximately a 15 to 20% increase in accumbal glutathione levels. Together, these findings convincingly demonstrate that glutathione concentrations in the nucleus accumbens do not merely correlate with motivated effort, but play a causal role in sustaining reward-driven behaviors. This study not only tells us something about the biochemical underpinnings of motivation, but also identifies glutathione enhancement as a potential therapeutic strategy for conditions characterized by motivational deficits. The influence of glutathione extends beyond antioxidant protection. The authors suggest that manipulating glutathione levels may directly modulate motivation by reshaping glutamatergic signaling in the nucleus. Glutamate is a neurotransmitter used for excitatory signaling in the brain and must be tightly regulated to ensure precise neuronal communication. This neurotransmitter balance is linked to glutathione metabolism. Not only is glutamate a precursor required to synthesize glutathione, but the process of synthesizing and recycling glutathione can also influence local glutamate availability in the brain. Moreover, treatment with N-acetylcysteine appears to affect glutamatergic signaling through multiple pathways. Importantly, NAC-derived cysteine, that's two cysteine molecules bound together, also interacts with glial cell transporters, specifically cysteine glutamate exchangers, influencing extracellular glutamate concentrations. By modulating extracellular glutamate levels, NAC indirectly impacts the strength and the frequency of glutamatergic synaptic signaling, specifically in neurons of the nucleus accumbens that control motivated behaviors. While previous studies on NAC's impact on glutamate signaling primarily showed general short-term effects, the Zalakora study showed longer-term cell-type specific adaptation. The sustained motivational benefits from increased glutathione likely involve a combination of potent antioxidant protection, the acute modulation of glutamate availability, and more enduring reshaping of synaptic connections within the nucleus accumbens. So, how can we support our body's ability to make glutathione outside of some of the obvious recommendations like getting plenty of sleep and exercise? Let's start with the compound used in the study that we just went over, N-acetylcysteine. NAC is one of the most widely studied and clinically used supplements for raising glutathione. It works by providing cysteine, a rate-limiting amino acid your cells need to synthesize glutathione. Importantly, NAC crosses the blood-brain barrier and has been shown to raise glutathione levels in the brain in both animal and clinical studies, with the clinical studies mostly focusing on populations with brain disorders tied to lower neuronal glutathione levels, like Parkinson's disease. Now, most of those studies in humans use NAC via intravenous administration. Oral NAC has relatively low oral bioavailability. However, there are human studies showing oral NAC raises circulating glutathione levels in people with lower than average circulating glutathione. Most human studies use between 600 and 1800 milligrams per day divided into 2 or 3 doses for the purpose of raising glutathione. The higher end of that range has been shown to be successful. Some protocols go up to 2400 milligrams, but that's typically reserved for clinical settings. The standard range, 600 to 1800 milligrams, is very well tolerated over long-term use. Now, NAC can be taken acutely, for example, before a demanding cognitive task, but its more robust benefits on brain chemistry, oxidative balance, and motivation tend to build with daily use over several weeks. Even though NAC supplies cysteine, it's one piece of the puzzle. Glutathione is a tripeptide made from glutamate, cysteine, and glycine. In some people, like older adults, glycine becomes a second rate limiting factor. So even if you take enough cysteine through diet or supplementation, your body might not be able to synthesize enough glutathione if glycine levels are low. There is data that has shown that a combination of glycine and NAC gives your body both of the limiting precursors it needs to restore glutathione. A pilot study in older adults published in Clinical and Translational Medicine used 1.5 grams of NAC and 1.5 grams of glycine taken twice daily. That's 3 grams of each per day. This combo in the industry is called Glynac. After 24 weeks, participants didn't just normalize their glutathione levels, but they also saw improvements in cognitive function, mitochondrial function, insulin resistance, muscle strength, walking speed, and even circulating BDNF levels. When Glynac was discontinued, many of those gains reversed within 12 weeks, suggesting that these effects weren't incidental. They were directly linked to sustained improvements in redox status and mitochondrial resilience. Compared to NAC alone, the combo of glycine and NAC might be a more complete intervention, particularly in aging populations or individuals with high oxidative burden. NAC on its own still holds value, especially in younger adults or in cases where glycine status is adequate. But for restoring full intracellular glutathione and unlocking the broader benefits associated with redox balance, GlyNAC does appear to be the more comprehensive approach. Now, some people do ask about just taking glutathione directly, but the truth is oral glutathione has very poor bioavailability. It's mostly broken down in the gut, and it cannot enter a cell as preformed glutathione. It needs to break down and reassemble to do so. Liposomal glutathione and IV formulations can raise plasma levels temporarily, but they haven't shown the same depth of effect as NAC or GlyNAC when it comes to supporting motivation, cognitive stamina, or metabolic health. The latter just have more and better data to this point, and as a bonus, they're also cheaper. Those are some more direct ways to boost glutathione, but what about some indirect ways, specifically through foods and compounds that help your body maintain a strong antioxidant defense? One of the best researched examples of this is blueberries, specifically containing anthocyanins. Blueberries, especially wild blueberries, are particularly rich in anthocyanins and other flavonoids. These are not just general antioxidants, they directly stimulate pathways in the body that increase glutathione levels. They do this partly by activating a cellular pathway called the NRF2 pathway, which helps ramp up your body's own antioxidant defenses. Specifically, it raises levels of frontline antioxidant enzymes, one of those being glutathione peroxidase enzymes, which use glutathione to neutralize free radicals. Animal studies have shown that blueberry polyphenols, particularly anthocyanins, increase glutathione levels in the brain. These studies suggest beneficial effects at around 30 to 60 milligrams per kilogram of body weight of wild blueberry extract. This dose significantly increased whole brain glutathione levels by about 28%, and that equates to a human equivalent dose of around 350 milligrams of concentrated blueberry polyphenols per day. That data does line up with the available clinical data in humans. Human trials have used various forms of blueberries, from fresh berries, about 1 cup, that's 100 grams daily, to concentrated juices equivalent to around 230 grams of fresh blueberries, or even purified extracts standardized to about 100 to 300 milligrams of anthocyanins per day. A recent human trial from 2024 demonstrated that an acute dose of 222 milligrams of wild blueberry extract significantly enhanced cognitive performance and attention in older, healthy adults. This was particularly true during times of the day when cognitive function naturally dips, like after lunch. Participants who took the blueberry extract maintained faster reaction times and better sustained attention throughout the afternoon compared to a placebo. In short, blueberries, particularly wild blueberries or blueberry extracts, are dense in anthocyanins, offer meaningful support for brain health and cognitive function primarily through indirect boosting of glutathione and antioxidant enzymes. 5.5 grams of just blueberry powder or extract standardized to 300 to 500 milligrams of anthocyanins has shown to provide these cognitive benefits, particularly during periods of high oxidative stress or cognitive demand. Now, let's briefly touch on 2 more indirect ways to support brain glutathione. Alpha-lipoic acid, or ALA, is a unique mitochondrial antioxidant that not only protects cells from oxidative stress, but also generates glutathione by activating the NRF2 pathway, the same transcription factor that drives the expression of enzymes responsible for making glutathione. It does this by helping NRF2 disassociate from another protein called KEAP1, and that allows NRF2 to translocate to the nucleus of a cell, thereby stimulating antioxidant response element. Studies in animals consistently show that ALA raises brain glutathione levels, especially in the hippocampus and prefrontal cortex. Human doses typically range from 300 to 600 milligrams per day, though they do go up to 1,200 milligrams per day, and that's used in trials for neurodegenerative conditions. Because ALA has a short half-life, splitting the dose, say 600 milligrams twice daily, can help maintain steady-state circulating levels. Then there's a compound called sulforaphane, the active compound in broccoli sprouts. Sulforaphane is a potent activator of NRF2 and has been shown to increase both blood and brain glutathione levels, even in healthy humans. One study using approximately 40 milligrams of sulforaphane per day for just 7 days significantly raised glutathione levels in the thalamus. That's a brain region involved in alertness and information processing. Other data has shown metabolic benefit as low as 1 to 10 milligrams per day, depending on body weight. These low quantities are likely attainable via raw broccoli or cruciferous vegetable products, although higher doses are likely needed for cognitive benefit. Sulforaphane doesn't supply building blocks for glutathione. It instead ramps up the system that makes and recycles it. Think of it as turning up the volume on your cellular antioxidant machinery. Animal studies suggest that sulforaphane also helps preserve glutathione in regions like the hippocampus and the substantia nigra, while also reducing inflammation and oxidative stress in the brain. Clinical trials are still emerging on sulforaphane, but in both aging and psychiatric populations, sulforaphane has shown potential to enhance cognition, especially under stress. So while ALA, alpha lipoic acid, improves glutathione by fueling mitochondrial resilience, sulforaphane boosts the system itself, making more glutathione across multiple brain regions. Together, these compounds illustrate a theme. The brain's ability to sustain motivation and mental performance depends in part on antioxidant capacity, and glutathione sits at the center of that defense. Motivation isn't just a mindset, it's a metabolic process. When your brain pushes through effort, especially in high stakes or high demand situations, it creates oxidative stress. And glutathione is, among other things, what helps keep that stress in check. It's the buffer that lets your neurons keep firing when things get mentally hard. The nucleus accumbens, the part of your brain most responsible for effort, drive, and reward, runs hot when you're pushing through a task. That region burns energy fast, and without enough antioxidant protection, it starts to fatigue. Glutathione is what allows it to keep going. We've seen that people with higher levels of glutathione in this brain region are better able to sustain motivation. They don't crash as quickly. Their brains are more resilient. So what is it that we can do? Well, we don't need to chase exotic compounds. You can support glutathione naturally, starting with your diet. Blueberries, broccoli sprouts, and high protein foods like eggs and fish all feed into your body's antioxidant system. These aren't just good for you foods, they actively help your brain buffer stress and stay sharp. If you want to go a step further, supplements like N-acetylcysteine or glycine, alpha lipoic acid, or sulforaphane can give more targeted support, but the core idea remains the same. Motivation depends on energy, and energy requires protection. Glutathione is that protection. So if you find yourself running out of mental gas, it's worth asking not just, am I tired, but is my brain equipped to stay in the game? Support your antioxidant system, and you just might find your motivation lasts longer than you thought possible. Thank you for joining me today on Daily Value. Stay motivated and stay healthy.